I'm from Denmark and got a DVM from the University of Copenhagen and worked in mixed practice and equine practice, and then met Henry Stampley from Large Animal Medicine and a sports medicine conference, and eventually came to Canada to do internship and residency. The work on umbilical cord blood derived was first done in Canada, and we had the first report on that in 2007, which gained highly accessed status because it was downloaded more than 3,500 times in the first 2 weeks after publication. So obviously the goal here is to try and find new treatment modalities for diseases and conditions where we don't have good treatment modalities today. So the focus of my lab is on joint cartilage repair. We hope that this research might assist in horses with developmental conditions to maybe bring them to market and allow them to perform as the breeder intended. And for horses who are already working, we are hoping that we can salvage their performance and reduce the number of horses undergoing premature retirement and loss of training days due to joint cartilage disease. The goal of our research is joint cartilage therapy, and not degenerative joint disease, so focal cartilage defects, to try and intervene early in the process and restore that cartilage so it doesn't progress to fulminant osteoarthritis. And whether that will actually, it could happen in a number of different ways potentially. We don't know how these cells really exert their function in vivo. It could be that we can find a way to make them integrate and create new tissue and actually be part of that newly formed tissue. Or they may have a more secretory function where they secrete signaling molecules that then govern the microenvironment, both the immune environment and the inflammatory response in the joint, but also maybe help in recruiting resident cells from the surrounding tissue to actually do the repair or creating new tissue. In young animals, it could be osteochondrosis, so a developmental condition of the joint cartilage that leaves either joint mice or a bone cyst that you want to try and repair. In older horses, it's kind of a sequelae to microtrauma or the continued work and load on the joint, and then there are areas that are getting affected and you create focal cartilage defects. You can basically get what we call mesenchymal stromal cells from any tissue in the body. So in any tissue in the body there are these resident cells. And in my lab, that's what we work with. And those cells we isolate quite simply, utilizing their ability to adhere to plastic. So we take these cells and we put them into a flask like this, and a number of the cells will stick to this plastic surface and then grow and expand, and a number of cells won't. They'll be floating in the media, and when we replace the culture media, we get rid of those cells. So over weeks, we get a more homogeneous cell population out of this flask that we then, through chemical molecules, can direct towards cartilage cells or bone cells. So what we are working on now is a better way of differentiating or making these naive cells into cartilage cells in the lab so that we would have enough cells for therapy. Naive cells, or that, what I mean is the undifferentiated cells. So that kind of brings us to the discussion about stem cells and what is a stem cell. And in my opinion, there are no stem cells in equine medicine today. There are cells that we can differentiate into certain tissue types, but they don't possess the hallmark of a stem cell that you can expand them indefinitely in the lab. So I know a lot of people refer to these cells as stem cells, but I would caution on that and say that, yeah, I'm sure we can find stem cells in horses and derive equine stem cells, but we're not there yet. So the work that's done so far is more on what we call progenitor cells. So it's not this very potent stem cell that can make any tissue in the body. But we might not need that either, because from a clinical point of view, if you have a stem cell that can make any cell in the body, one of the criteria for a stem cell is also that they can form tumors. So as a clinician, that's a concern, because if you have such a potent cell, how do you control it in a patient and not end up with cancer or tumors? So if you can have a cell that can form cartilage, and that's what you want to repair, that might be all you need. Because when you have a defect, obviously you have normal healthy cartilage surfaces on the side, and then you have bone underneath. And it's a problem getting all that well integrated. So I think what's being done today with different matrices and scaffolds and cells in there, it may turn out to be a solution, and there are other scaffolds being developed all the time that are more sophisticated. An alternative which we are pursuing is to make tissue engineered constructs that has a top layer of cartilage and a bottom layer of bone. So you basically have this plug of cartilage and bone that you can then put into the hole, or maybe you need a number of the plugs to fill up the defect. And that technique has been pioneered by Mark Herteg here at OVC, mosaic arthroplasty. And that might allow for better integration between cartilage and bone. People today are mainly harvesting it from fat tissue, and from bone marrow aspirates. So, and I work with cord blood derived cells, and there are other people working with that as well, and from other tissues. But the two main cell sources at the moment in the horse is either from fat tissue or from bone marrow aspirates. The alternative, what's being used today, is through injection of cells and scaffolds together. And it's appealing because you can do most of this through the arthroscope. So it's minimally invasive, and it's relatively quick, and it's much faster than having to develop tissue engineered constructs in the lab, with also possible associated risk of infection and contamination, because you have manipulated it in the lab for a long period of time. So those are kind of dual tracks going on at the moment. We have observed this donor-donor variation, as we call it, that cells from one animal doesn't behave the same as cells from another animal. So when we have, for example, different cultures from cord blood from one horse would be very good at forming cartilage in the lab, and cells from another horse would be not so good. So that's obviously a concern if you want to use these cells in that horse later on. So what we are working on now is it possible to use cells between individuals? So if you screened these cultures and found the ones that were really good at making cartilage, could you use them for other individuals than the one you got it from? So that's one thing that we are investigating now. The other thing is that maybe you can identify cells in the culture that have good potential of forming cartilage. So maybe in the culture that we now describe as being poor at forming cartilage, it's a heterogeneous cell population. So that means that all the cells are not the same. So maybe in that culture we can find cells that are actually good at forming cartilage. They're just not in high enough numbers at the moment. But if we could find those cells and then selectively expand those, then suddenly we would have cells from that individual that were good at forming cartilage. And you wouldn't have the concern of an immune rejection because it's cells from that same individual. in our lab at the moment, we're basically ready now to do in vivo studies in the horse, but we would like to get a little bit more mechanistic insight into what the cells are doing in the live animal. So instead of just injecting the cells and then measure the outcome, the clinical outcome, whether they are less lame or whatever you're looking at, we would like to know what happened to the cell. So in order to do that, we need to label the cells so that we can identify them and discriminate them from the host cells that were already there. And that has proven a bit challenging because a lot of these particles and methods that you can use actually turns out to be toxic to the cells. So they might change their normal biological behavior. I've been extremely privileged with funding. My PhD was funded through the Danish Research Council to study 3 years abroad here at the University of Guelph. And during that time we got additional operating money from Equine Guelph and their partners, which we're very grateful for because it really helped put this research on the international map. And it later allowed us also to obtain funding from Grayson Jockey Club in Kentucky. And during my PhD, we were also contacted by a company, BioE, in Minnesota to do a project together with them, and I continue to work with them now. And now currently I'm funded through the Danish Research Council again with a 3-year grant. And funded through Equine Guelph and their partners, and working together with another US company, Centrix Animal Care, who has a hydrogel for wound repair that we are gonna work with as well. And just last week we got funding from Morris Animal Foundation in the US to start work on canine stem cells, these, as I've said before, these induced pluripotent stem cells. So that project is in collaboration with Professor Dean Betts, Professor Tom Gibson here, and Professor John Lamar.